Welcome to this morning's tutorial on some higher biology data handling questions to help you with the exam coming up next Tuesday. So what I've got here today for you to look at is a graph. Okay, there is a lot of information on this graph, but how we are going to approach it is by doing three simple things, right? So the three simple things we're going to do is I'm going to take a random colour here. I'll go for the brown, and you're going to be able to break this question into three different parts, right? So part one is going to be, sorry, I'll just make that a little bit smaller. Try that. Part one, okay, is going to be the context of the question. Part two is going to be actually sorting out the data on the question. And then finally, part three is going to be looking at the question itself, right? So when you come across this type of question in an exam paper at a higher level, or higher human course as well, you will know exactly how to approach it. So to answer it successfully, take three different coloured highlighters with you into the exam and make sure that you have them readily available to mark on your paper, right? So the first part is, before we get on to sorting the data, is looking at the context. So what is this graph? What is it all about? Okay, we've got three different sides, okay, with three different readings. Okay, so effectively I call it X, Y, Z graphs, right? But what we've got is the context, and the context is the graph below shows the rate of potassium uptake and glucose breakdown by muscle tissue in solutions of different oxygen concentrations. Okay, so let's sort out the information. We're going to look at each scale and look at the label units attached to each of them. What we're going to do first of all is go into the x-axis right along the bottom, and I'm going to take one specific coloured highlighter, and in this case I'm going to make it, we'll just go with red that I've already got highlighted, and all I'm going to do is highlight the labels and units at the side, snap that so it's clean, and then we'll do the same again. And you'll also notice that alongside this we've got a solid black line, right? So we've got the labels, we've got the units, and we've also got the corresponding line onto the graph. The next thing I'm going to do is I'm going to highlight the scale. So I'm going to take that up there. Alongside that, because we're dealing with the solid line, I'm going to take the highlighter and roughly highlight the solid line as it's placed upon the graph, right? So why I've done this is because whenever I'm going to be asked a question about potassium ion uptake, okay, or if I'm going to be asked a question on potassium ion uptake, I'm going to get my reading from this dark solid line, but I'm only going to take the reading from the scale on the left hand side, which matches up to the highlighter, okay? So a common thing that can go wrong is you might see this point here actually touches the scale on the right hand side. So a common mistake that's made is, for example, if you're asked to read what this point is, you might see that as being, well, it's going to be 15.5 in this case as the increments go up in 0.5, which would be completely wrong, because even though it's touching the scale, it still needs to be read from the highlighted red axis, right? It takes away a bit of the confusion. Next up, we're going to take up another colour. We'll go along the bottom, we'll do percentage in oxygen solution. Snap that so it's clean. And then we're going to go along the bottom So if ever we're asked about oxygen solution, we know what scale we're going to use for and where we're going to get our reading from. Okay, so they go from the bottom of the graph all the way up to the top to meet readings for what's on either side. Right, so that's pretty straightforward. So we'll go on to our final colour and we'll make this one, what will we go for? We'll go for purple, okay? Take a highlighter, as same as before, snap that till it's clean, and then go here, and then the other information you get from this as well as the labels and the units is the type of graph that is being used. So go back to the highlighter, highlight the dashed line, and then run the highlighter up the scale, and then highlight the actual dashed line. Okay, so if ever we're going to be asked about units of glucose, we're going to go to the dashed line and then we're going to take a reading from the right hand side of the graph, okay, not the red at the left hand side, because that is a completely different scale. It's not relevant to the units of glucose broken down. Right, that completes stage 2. Okay, so stage 2 is now complete. We have sorted all the information in the graph and we know exactly what story the graph is trying to tell us. And now we go into stage 3, which is looking at the actual question. When we look at the question, we're looking for hints. Okay, so how much glucose is broken down per minute when the oxygen concentration is 1%, right? So the key things that I'm picking up here is glucose broken down per minute, and also oxygen concentration at 1%. Right, so with that in mind, I'm going to, what colour I'll go for here? I'll go for this nice blue. If it's at the oxygen concentration of 1%, it's going to be red from here. So what I'm going to do from here is I'm going to take my pencil, okay, in this case it's a blue pencil, and I'm just going to run that line all the way up to the top. Okay, and that's going to allow me to read the actual result from the glucose being broken down. Now, the glucose being broken down is here. Okay, so I'm only dealing with the highlighted purple sections of this graph once I've sorted out the information on it. Okay, so what we've got is we're just going to go all the way up this line until we meet the dashed line, which is going to be roughly there. So I'm going to take my pencil, take the blue pencil, and I'm going to put, in fact I'm going to make it red, okay, and I'm going to put that X right there, and remember it's the purple highlighted line, so it's going to be red from the purple scale. So going back to my pencil, I'm going to take that and then just run it along here, and the answer to this question is going to be right there on that scale. Okay, so looking at the answers, I mean, I haven't even touched on the answers just yet, but having an idea roughly where this point is in the graph is quite obvious. Okay, so I can see that it's somewhere between 5 and 0. Looking at the answers, the options I've got is A, 2.5, B, 6 units, C, 10 units, D, 24 units. So looking back at it, it's roughly halfway between 0 and 5. Now, on the exam, you're going to take a ruler and be very, very accurate, but I can tell simply by looking at this that the value is between 0 and 5, and it's roughly halfway. The only value that's an option in the answers is going to be A, 2.5, because the rest, okay, are over 5. So it can't be 6, it can't be 10, it can't be 24, leaving the only option remaining as A. Okay, so that's pretty straightforward how to do that question. So before I go on to the next one, if you just hit the like, subscribe, or even if you want to ask me questions to do through the live stream. I should be live streaming most of Monday, So if you have a particular question that you want me to answer, then please do get it in, and I'll do it for you on Monday. Okay, very, very important day because it's the day before the actual exam. Okay, so thanks for joining me and watching this, and I'll see you in the next clip.